Hello and welcome to SAM online class. This is Dr. Subrat Kumar Mahanti. Friends, today we shall discuss about in situ hybridization. In situ hybridization is a powerful technique that is used to detect and localize nucleic acid sequences within entire tissue, portions or sections of tissue, within cells and also in circulating tumor cells. The technique of in situ hybridization was invented by American biologists Mary Lapar and Joseph G. Gall. There are different types of in situ hybridization. One of the hybridization is fluorescence in situ hybridization. Fluorescence in situ hybridization is a type of in situ hybridization that uses fluorescence level probe. Otherwise, the basic principle of different types of in situ hybridization remain the same. So, by in situ hybridization, we can detect and localize specific nucleic acid sequences within cells, in circulating tumor cells, within entire tissue, even within portion or section of tissue. In situ hybridization has several applications. For example, in clinical cytogenetics, also in gene mapping. This technique is also used to study tumor biology and chromosome evolution. So it is a powerful technique. The process of in situ hybridization is based on using a suitable probe. What is probe? Probe is a oligonucleotide that can be labeled radioactively or non-radioactively. So by using labeled probe, one can easily detect the presence of specific nucleic acid sequence within cells or within tissues due to hybridization. The process of in situ hybridization uses different types of probes. The probes can be a double-stranded DNA, single-stranded DNA, or it can be a riboprobe, or the probe can be a synthetic oligonucleotide. So the probes that are used for in situ hybridization are usually labeled. Then only we can detect the presence of specific nucleic acid sequence. The probes can be labeled radioactively or non-radioactively. For example, by using fluorescence. Radioactive labeled probe can be prepared by using radioactive isotope, for example, hydrogen 3, sulfur 35 and phosphorus 32. Similarly, probes also can be labeled radioactively, for by example by using digoxygenin, biotin and also by fluorescence. Like other technique, the technique of in situ hybridization also involves several steps. First step is preparation of slide. For chromosome preparation, methanol and ether clean slide is sufficient. Whereas for tissue section, polylysine or glutaraldehyde activated gelatin chrome aluminium slide is preferable. So first step is preparation of slide. Second step is sample collection. For in situ hybridization, the sample must be collected carefully in order to preserve morphological integrity. The tissue sample should be collected rapidly and it should be properly fixed. For fixation of tissue sample, different type of fixatives are used. For metaphase chromosome spread, one can use methanol and acetic acid as a fixative, whereas for paraffin embedded tissue, formalin fixation is more suitable. After sample preparation by using a suitable fixative, next step is embedding and section preparation. The tissue sample is usually embedded in paraffin or OCT for long-term storage or for sectioning by microtome for further uses. This is third step. Fourth step is permeabilization. Permeabilization is a process that is carried out in order to increase the permeability of the tissue sample so that the probes will easily infiltrate into the sample in order to reach the nucleic acid sequences so that hybridization can occur and the sequences can be detected. Permeabilization of the sample can be done by different methods. For example, by using the enzyme proteinase K and also by using 0.2 m hydrochloric acid and also by using various detergents like Triton X100 and sodium dodecyl sulfate. So this permeabilization is a process that is necessary to increase the permeability so that the probes can easily infiltrate into the tissue sample in order to reach the nucleic acid sequence. Fifth step is pre-treatment and pre-hybridization. Pre-treatment and pre-hybridization is a step that is carried out to lower background noise because other molecules are also present in the sample, so they may interfere in the process of in situ hybridization. In order to minimize the interference and to minimize the background noise, pre-treatment is necessary. Pre-treatment can be performed by different processes. One is by inactivating endogenous enzyme. The enzymes that are already present in the sample may interfere with the process of in situ hybridization. So, the endogenous enzymes must be inactivated before hybridization. Second is RNase treatment. The sample can also be treated with the enzyme ribonuclease. Ribonuclease treatment degrades the RNA molecules that are present in the sample, so the background noise will be lowered. And also by the process of pre-hybridization, the background noise can be lowered. Pre-hybridization is a process in which the section or the sample is treated with a hybridization mixture that contain all the components except probe. Sixth step is hybridization. It is the most important step. The process of hybridization involves the treatment of the prepared sample with probes. As you know, probes are nucleotide sequences that are labeled. By using labeled probe, we can easily detect the presence of specific nucleic acid sequences within cells and samples. So, in the process of hybridization, the sample is treated with a suitable probe so that annealing will occur between the probe and the nucleic acid sequence. Annealing occurs between probe and the nucleic acid sequence due to complementarity of the base sequence. Seventh step is washing. After hybridization, washing is performed. Repeated washes are performed in order to remove excess or unbound probe that may be present in the sample. This is about washing. Washing is performed by using a suitable solution in order to remove excess and unbound probe. Last step is detection. The method of detection that is used to detect the presence of nucleic acid can be of different type. For example, if the probe is radioactively labeled, then it can be detected by using photographic film or photographic emulsion. And if the probe is fluorescence labeled, then other methods must be used to detect the presence of nucleic acid sequence within sample. So friends, in situ hybridization is a powerful technique that is used to detect the presence of specific nucleic acid sequences within intact cells, tissue sections, even within entire tissue or in circulating tumor cells. So it has several applications. So friends, this is about in situ hybridization. Please subscribe my YouTube channel known as Sam Online Class. Give your feedback. Thank you very much. See you again. Bye bye.